Yeah, we're starting live stream. Okay. All right, I guess we are live now, and probably we can get started, Ercument. All right. Hello, everyone. It's my pleasure to introduce Ercument today. Ercument is an associate professor at Bilkent University in the computer engineering department. He's also a visiting professor at EPFL currently, where he's doing his sabbatical, I assume. So maybe I can give more information about him from his bio. So his research is focused on using machine learning techniques to uncover the genetic basis of complex disorders. You're going to hear actually more from some of his works actually today. To achieve this goal, he works on a variety of research questions, such as detecting structural variations, assessing gene risks, developing generative models for drug design, and implementing privacy-preserving distributed methods for sharing biomedical data and training models. So Ercument's research has been funded by several institutions such as NIH, Simons Foundation, and TÜBİTAK, and he has received research incentive grants from several institutions, including the Turkish Academy of Sciences, Health Institutes of Turkey, and Science Academy of IEEE Turkey. He's also, I guess, one of the masterminds behind the Hercules paper, where we published and also developed Apollo afterwards. He's been a really nice collaborator when I was doing my master's at Bilkent. So yeah, so take it away, Ercument.
Okay. Thank you very much for the kind introduction, Jen. Also, thank you very much for being here. So today I'm going to talk about a specific part of our lab, which focuses on detecting copy number variants, which is a type of structure variants, type of mutation, basically. And I'm going to talk about what this type of genetic variation is, what are the challenges with detecting those, and our solutions to address this. Okay, now it decided not to work.
Maybe we can click on just one.
Okay, I can just go. Okay, now it works. All right, so structural variation means basically the differences among individuals or within an individual with respect to some subset of the genome. So usually the genetic variation that we consider is just point mutations, changes of a single or just a couple of nucleotides, but structure variation is basically larger, dealing with larger variation, some larger portions of the genome being different among individuals. So it's basically from a couple of hundred nucleotides to like a million nucleotides, like a megabase change. These are some examples of such changes. So you might so this is the reference genome, like the normal human genome that we consider, and you might, so this is the person that you consider. You might have like a portion of the genome deleted, or you might have a new novel sequence inserted. Some sequence called mobile elements can jump from position to position, and it might get stuck in a new position in the genome. So this is another type of an insertion. You might have, so for the duplications, meaning, it's another type of insertion. You might have a certain portion of the genome duplicated and inserted right next to the same position. So it might be doubled, or you might have some interspersed duplication where you get this chunk and then it gets inserted to somewhere else. So these are called basically copy number variations. As we have two chromosomes, you would expect a person or a sample to have, a human sample to have like 2 copies of each sequence. But in this case, for instance, you would have, for instance, if one chromosome has 1 copy and then here you have 2 copies, the copy number in this case is 3. So this, and then we have other types of structural variations. So these are called balance rearrangements. You might have a sequence gets inverted and inserted the same position. So it's balanced because you have the same number of nucleotides, but the order is changed. Or you might have a portion of the genome gets ripped out from a part of the chromosome and gets inserted to gets stuck in somewhere else. So this is called translocation. So here today I'm going to talk about these types, which are called copy number variations. So these structure variations are individually very rare. You would expect like 15 to 20 million base pair changes per person. So it's less than less than 0.001 per person. A genome is like 3 billion nucleotides. But it's very common collectively. So this is like a map of all chromosomes, and then in 2008 they have cataloged the set of copy number variations across the human genomes. You see that it's almost everywhere. But of course, there are conserved regions as well. And this is good for kind of driving the genetic diversity among humans. That that's what makes us different, of course. But it might also lead to serious diseases, like for instance, deletion causing neurodevelopmental disorders. If it affects a certain, a conserved region of the genome, meaning it's very important for function and survival. So duplication, for instance, might lead to schizophrenia, psoriasis, and so on. So these it's really important to understand and detect structural variation and copy number variations to study disease, and that's what we are trying to do. So there have been a large effort on doing so. International projects trying to catalog all types of copy number variations, and this paper from 2015, for instance, in a single study, they were able to find more deletions than we have ever cataloged. And then you see the length distribution for different types of deletions, duplications, insertions, and so on here. So my point so far is that copy number variations are an important source of heterogeneity among humans, and then also it might have implications in human health. So it's important to detect them, and there has been a large effort. So here you have the size of the variants. We have single nucleotide changes. So the other extreme is having an extra chromosome, or losing a chromosome, maybe trisomy or monosomy. And then copy numbers are basically in between. And there have been many methods to detect them over the years. One of them being, for instance, karyotyping. So you basically, under the microscope, see the chromosomes of the person to understand the changes. But this could only detect, for instance, changes over 1 megabase. So it has to be huge, maybe like an extra chromosome. But with high throughput sequencing, this changed. So now basically in theory, we are able to detect all types of genetic variations, including copy number variations. And you can do the high throughput sequencing for this purpose over 2 data types. The first is whole genome sequencing, where you sequence the full genome of the sample. And the second one is whole exome sequencing, where you sequence only the coding parts of the sample. And then this has quite advantages, so I'll come back to that. There are many methods to detect copy number variations. The first one could be a read pair analysis. So you have the reads are paired in this. So for instance, in Illumina, and then you map them back. So you know the expected range between these two reads, and then if it is larger or smaller, then you conclude that there might be an insertion or a deletion in this region when you align it back to the reference genome. The second one, which we are interested in, is the read depth analysis. You get your short reads, or it could be even long in this sense. You map it back to the reference genome, and based on the coverage, if you see like a certain decrease in this signal, so I think you should appreciate that this looks like a signal. If the signal goes down, then there's a possible deletion in this region, or if the signal just has a peak over here, maybe there's like an insertion. There's also split read analysis. So once you map a read back to the reference genome, if the aligner allows you, then it might tell you that this read is split, meaning in the reference there's like an extra region whereas it's lost in this person. So if it's a large region, then you might conclude that there's a deletion for this person in that region. And final local and de novo assembly, basically to find, aligning short reads back to the genome is problematic because the human genome is quite repetitive, and the mapping is ambiguous. So you might easily miss some insertions or deletions because of this ambiguity. Instead, you do like a de novo assembly and map it back to find such regions. So these are like 4 techniques to detect copy number variants. There are many problems with detecting copy number variants, unfortunately. First, no technology is comprehensive. So here you see 3 different technologies and the copy number variants detected using those on the same 5 individuals. And then these are relatively large copy number variants, but everybody says something else, as you see in this 2010 paper. No technique is also comprehensive. So this is the 1000 Genomes Consortium paper. With different techniques, you were able to detect a different range of copy number variants, and then they hardly agree. So it means we don't have like a one-stop shop to find all the copy number variants. And this is the statistics from that paper. So we have different techniques: read depth, parent, split read, and assembly-based methods. So they try them all. And then here you see the number of structure variation calls, some of them, and this is the number of validated calls. So if you look at here, the read depth is quite advantageous. For instance, the CNV method is able to find so many copy number variants, and it has a high number of validated ones with a relatively low false discovery rate. But see, the numbers are still quite problematic. And then this is on whole genome sequencing data. Yes.
So do these numbers still hold true for current techniques? Because like this, from let's say 2010 until now, are the splits between decisions of different tools so
yeah,
wide still?
Yeah, it's widely speaking, it still holds. There are other methods. I was on a call yesterday that they were doing this. Still, they consider this problem as an unsolved problem, and then for certain regions, the sensitivity and specificity is still quite a big problem. So with the increasing read depth, you might solve this to a degree, but still it's an unsolved problem. Yes. All right. So these numbers I showed you is not good, but it was relatively okay with 14% this false discovery rate. And then this was obtained on whole genome sequencing, where we sequenced the full genome of the sample. Now, I want to compare the whole genome and whole exome-based sequencing for this purpose. So, in that paper, it was 14%. In the paper of CNV, they report like 5% false discovery rate. So it's relatively acceptable for this tool, which is considered, widely used in the domain. But if we were able to, if we wanted to use whole exome sequencing for the same purpose, the false discovery rate goes up to 60%. So this is highly unacceptable, as you might imagine. So the reason is that it comes with a uniform read depth distribution over the full genome with very nice properties. Whereas I'll show you an example in whole exome sequencing data, this read depth distribution is quite non-uniform. But why do we want to do it over whole exome sequencing data then? So the reason is with whole genome sequencing data, you have the data sizes are quite large. This is 100 gig on average, and then this is like 3 gigs. This is more expensive. It's around $1,000 now, and recently got below. This is much cheaper compared to whole genome sequencing. This is less abundant. For instance, in GLOMAT, when I checked, it was like 15,000 whole genome samples, whereas now the whole exome sequencing is quite abundant. So this is a data set that basically catalogs the exome sequencing and genome sequencing data obtained through studies over the world. So it would be really great if we were able to do it, even at this, with this level of false discovery rate, using whole exome sequencing data, because it's widely used in clinic because of all these advantages, unlike whole genome sequencing data. I didn't even mention the time. This is much, much faster to obtain, of course. So this is one example from IGV, meaning I'm showing you a portion of the gene. Actually, this is a chromosome, and then the reads map to this chromosome. And here you see the read depth signal that falls onto this chromosome. It is the same sample using the whole exome sequencing data. So with whole genome sequencing, I see a very nice, uniformly distributed read depth. Whereas for whole exome sequencing, since I'm only sequencing certain parts of the genome, I see just peaks. So it's reasonable to say it's really, really hard to detect such changes in the signal using this peak data. So that's why this copy number variation on this type of data is not a good idea at this stage. So our research question is both of the words possible? Meaning, can we achieve this copy number variation detection performance obtained on whole genome sequencing data using whole exome sequencing read depth signal? So it's going to be fast, cheap, and so on. So the answer is probably no if we use only that noisy read depth signal, because more noise than probably the signal. But the answer is maybe if we were able to tie the whole genome sequencing read depth signal and the calls made that on that nice signal to the noisy whole exome sequencing based signal. And then the core idea is that we have hundreds of matched whole exome and whole genome samples in the 1,000 genomes data. So we have around 2,500. So I have both signals for the same person. And as I told you, we are able to do it relatively better at whole genome sequencing data. So if I am able to train a model which can learn the association between the whole exome based signal, read depth signal, and the copy number variation. So what we do is we obtain this noisy signal. This is the uniform signal, have the calls, basically labels on this better data, and then try to associate this noisy signal with this higher quality labels. So that's what we are trying to do. Any questions at this stage?
So what you're trying to do is to train. So the idea is to train on the whole sequencing, but then the inference is done on just the exome part, right?
Can you repeat?
So you train the machine learning method on the whole sequencing, and then the inference you do only on this limited.
That's true. Okay. Yeah. So I input the exome sequencing data, and the labels come from whole genome sequencing. So the labels are relatively more trustworthy than I would get using this noisy signal. So what if the model gets the noisy signal and learns to produce the high quality label? So that's the point. And then this high quality labels are obtained using high quality signal. Yes.
Yeah, I'll try to distribute it.
So if we base on the whole exome, it means like certain parts of genome we not take in consideration. And my question is, so the copy number variation happens in the encoding part of the genome?
No, it happens everywhere. But if you are already interested in the whole exome sequencing, then you are making the assumption that the condition that you are analyzing is related to the coding part, which is like the, in quotation, active part of the genome. So the non-coding part of the genome is basically the regulatory, and you lose some information, but you make the assumption that it's more likely to be affecting the coding part, which produces the transcripts and the protein.
Yeah, thanks.
But yeah, you might lose information.
Just one question, right?
Yeah.
I mean, detecting let's say the copy number variations can be also like associated to the genes somehow, and then the non-encoding part is that probably can have information as if the gene is on or off. Does it useful to detect that there is variation even without knowing about when or it's on or off, basically?
So your question is, if we knew the non-coding part, would we know more about the gene's activity?
I mean, like the coding part is the one that have the gene,
yes,
right? Where you can basically like how a gene is mutating.
Yeah.
But if you only focus on the coding part, mean you don't know when this if this parting is on or off, and how important, what are the application for this, like just detecting the variation without knowing if this particular gene is on or off.
Yeah, clearly it doesn't tell us, this DNA sequencing data doesn't tell us about the activity of the gene, if the gene is on, if it's producing transcripts or not. Yeah, so it might be like a secondary analysis probably. So you would do RNA sequencing analysis. So this one would tell you, you know, I see something weird with respect to this gene, and it could be good for diagnosing this person to start with. Or if you are doing like a cohort analysis for a disease, it might tell you if this gene is frequently affected by a copy number variation, like autism deletion, always in the same region, then you might have like a diagnostic criteria, also, you know, more research questions. Yes, it doesn't tell us if it's on or off technically, but then if you do more functional analysis, then you need to go for the RNA sequencing data problem.
Okay, thanks.
All right. So this is the paper published in Genome Research at the end of 2022, joint work with Furkan Özden, our master student in my group, and then also Can Alkan. So here what we do is the goal here is to learn to polish the calls of a third-party CNV caller. Basically, we use this, you know, there are many methods that does this on whole exome sequencing data, quite problematic. So we take their calls and learn to correct their calls, basically. This is a polishing algorithm. So what we do is we get the alignments, and then we calculate the read depth, which gives us something like this. And then we pass the read depth through a exome-based CNV caller. So over the shelf, we get it, it gives us calls. So only on those places where this method makes calls, we perform the correction. So for instance, it says del here, dup here, and so on. So for these regions, we pass it through a recurrent neural network architecture. A recurrent neural network, very briefly, is basically a type of a neural network which analyzes sequence data. So for every position on the read depth, it, you know, learns its importance or the embedding using the things that it learned previously. So for this position, for instance, it you know, also takes the read depth signal in the previous positions, and it does it actually in both ways. Basically, it learns the context for each read depth. So if there's like an ambiguity, hopefully we are going to be able to learn that. So we pass the read depth for each of these regions through a BLSTM layer, which is a recurrent neural network type, and this gives us an encoded feature for this read depth. So if we see an anomaly, for instance, it's going to be embedded here. And then we basically concatenate it with the prediction of this algorithm. Okay, this is the, you know, the transformed read depth signal, and then this is the prediction. We pass it through two fully connected layers, and it tells us for each and every one of these a probability to be a duplication, deletion, or basically no-call, normal. And the labels, as again, comes from a whole genome sequencing based method, which is not the ground truth. We don't know the ground truth in this case, but it is much better than what we have with the exome sequencing data. So it's like a semi-ground truth in this case. Using cross entropy loss, we learn to associate the embedded read depth signal with these better labels. So this is what the training does, and in of course, in the test time, inference time, we won't have these labels. We will just input the read depth, calculate the encoded feature, get the prediction of the, you know, third-party caller, just provide these to the model, and it's going to produce us the predictions for each position. So that's basically the idea. Yes.
So what is the reason we don't have a ground truth in this case?
Because it's really, you know, we have ground truth very limited because as I showed you, this is, yeah, I mean, genome is huge. You have many callers making calls, and they are not concordant. So the way to obtain a ground truth is basically to kind of get an intersection of their predictions, and then somebody has to sit there, look at the read depth, and conclude that this region is duplicated or deleted, and find the breakpoint. So it's a very, very difficult task to do. And it is, even though we have grand truth, I'll show you, for a very small number of samples, and it's not, we are not able to train a model for that. Okay. Checking time. Okay. All right. So this is the idea, and these are some results. So we are considering 3 methods here: XHMM, Conifer, and Codex2, which are well-known methods doing this on exome sequencing data. And the black bars show you the duplication, deletion, and overall precision after our polishing approach. And we improve it in all, basically in using all of the metrics and all of the comparisons. And in some cases we even kind of double the precision, so it works. And these are the results for 2 CNV callers that predict exact copy number variation. So the previous one was saying there is a duplication or not, deletion or not. This one is telling you there's a duplication and then I have 8 copies, for instance, or 10 copies. So this is control freak. Actually, Valentina is, if you know Valentina is the senior author on this. I think the senior author, yeah. So here so we have 20,000 copy number variation events, and here we have the absolute error for the original method and polish method. So if you see a bar over here, it means our absolute error is better. So it means, almost like 2/3 of the calls, we were able to improve control freaks results. And, some in like less than 1/3, maybe we are deteriorating their results. And this is the distribution of the copy number predictions of control freak. And this is the grand truth, or the semi-grand truth in this case. And this is what the count kind of changes. So the distributions of the calls are much alike to the grand truth. And as you see, it's around 2, which is the expected range, right? So we have 2 copies of each.
One question.
Sure.
Do you still provide the sequence that is in a particular location of the mutation that happens there? Do you provide information like, okay, I have a copy number variation here and the sequence is ATTGCCC?
Not the sequence, but we provide the read depth, meaning, we do the sequencing, I get these short reads, I map it back to the genome. If there's a duplication, I'm supposed to see a hike in the read depth, right? So we provide that signal, the depth of the reads that are mapped to that region. So if there is a deletion, I see no depth. Basically, it just goes down, right? Okay.
So, yeah. I'm asking because, like, for indels, some people take the information that you have there as a mutation and then try to associate that with some diseases.
Yeah.
For instance, okay, if it's a T, then you need to do that, and if it's a C, you need to do this. And then for copy number variations, is it really important to know also, there is cases where knowing the sequence exactly, okay, you have a repetition, but it's a repetition of C, a repetition of T. There is studies associated with such kind of stuff that if it's really important to know.
Yeah, so this only tells you that there is like an increase or a deletion at this region. And then you can of course, go back and get that sequence and see what's happening there. For instance, a certain exon is deleted for a huge gene. So what does it mean? Maybe it's like an isoform. So it can affect all those. So it's like a post process. You detect the region and then you get the region. But the sequence of the region is not an input in this case, which could be actually, yeah, which could make a difference technically.
But is this the case in most of these colors for copy number variation? They don't report to the sequence that is present, or it's just exception?
They don't use that information mostly. Now that I think, they use the read depth. The ones that we use all just use the read depth. What might matter is that the GC content of the region, for instance, might make it more difficult for the caller to make a call. That's like a post analysis, but you can also input that to see, okay, maybe like pay more attention, higher GC content, maybe you are more likely to make an error. Yes, that could be like an input, but the full sequence itself is not.
Okay, great.
So this is CNV-kit, another method which we again have better results. So I'm not going to go into details, but we checked here the same sample, different sequencing platforms. Does our method work? Because you need to train a model for every sequencing platform. Maybe it doesn't scale, you don't have the resources. So the same sample using 4 different sequencers, 2 different brands, it still improves the results in all cases. So train a model using Illumina, I don't know, in this case, we were using HiSeq 2000 data, but it also works with HiSeq 4000 or NovaSeq 6000. That's what it means. Here we try to check, for instance, what if you don't have a decont? Our model name is decont, by the way, I didn't tell. So decont model trained on the CNVs obtained from a certain color, like XHMM, this third-party CNV color. And then a new one comes. Do you need to train a new model from scratch? You can, but even if you don't, you can borrow a model that's trained on XHMM. Still, is able to improve the results. So this shows using the XHMM model, I can improve the results of Codex and Conifer, but with a smaller margin. So now this is kind of important. To benchmark my method, I am still using the calls made on the whole genome sequencing data with this still, problematic but better methods with 5% post-discovery rate. So it's not the grand truth, but semi-grand truth. What if we had grand truth, as John asked? So we have grand truth for 9 samples provided by this paper, Chason et al. But in 9 samples, you cannot train a model. But we wanted to check if it conforms to what we have over there. So we got again XHMM Conifer Codex, made the calls, but our grand truth is different now. This human interference, like a human expert grand truth. And then again, we were able to improve the results, but as you see, our precision is much lower, 35%. If we use the grand truth calls, because it's probably relatively more restrictive, and then it is different than what this CNV-nator method does. Right. So
Ercument.
Yeah.
So do you think this is like an overfitting issue or something else just basically caused by some other structures in the other population?
Yeah, thank you. Good question. It's not an overfitting issue, but I trained the model using some decision-making criteria of this tool, but my grand truth is quite different now. Somebody else, some person really running multiple methods, cherry picking, maybe interfering with what calls are correct or not. Basically, they are correcting. So the model is trained on one data, tested on other data. It's very, expected to have, worse performance. But yeah, we have a fix for that. I'm coming. Hopefully. So we tried. I mean, the details here is not that important. We tried, even though this is not a somatic CNV caller, meaning for cancer, analyzing tumors. We tried this. Sometimes reviewers ask things. So we were. This is one problem. There are 4 regions that are validated. So we know the grand truth. There are CNVs in these 4 regions for these samples. So we have 16 tumors. We know where there is a duplication or deletion. So we run XHMM using exome sequencing data. It doesn't return any calls for regions 2, 3, and 4. So nothing for us to do. I mean, we cannot correct the call if there is no call, right? And then we were actually able to correct one call. So XHMM says on the tumor sample, I have a duplication. On the normal sample of the same person, I have a deletion. This is very unlikely. You would, either one would be normal, the other would be duplicate or delete. So we were able to correct this. But okay, so what? So it's it's nice, but this is also showing that the underlying, if the underlying method doesn't work as intended, and then we are kind of a dependent method, a polisher on these methods. So it's a problem. We cannot make new calls, which is a restriction. So, okay, one question we got from the referees was, okay, maybe XHMM is quite liberal, making calls for everything it sees, and then you are just making it more conservative, just, eliminating some basic thresholding scheme. That's what they claimed, and then we checked if that is the case. This was a good suggestion, actually. So this is this is a confidence threshold. Basically, what we do is we have 3 options: del, dup, and no call. We return the maximum probability among this, like softmax probability. So if it's like one of them is like 34%, that's the decision we make, whether a deletion, duplication, or a no call. What if we are more strict? Only make a call if it's like this is above a certain threshold, which could be like 0.5, 0.6, 0.7. So only make a call if it is you're really certain. And then we vary this threshold. So here you are making a call only if it is you are quite sure. And we did the same thing for XHMM, which was the best method in our benchmarks. So you would expect, if we are a basic, thresholding scheme, you would expect the performance to merge at some point, but it does not merge. We have the gap. Even if we are making a call, when we are really, really sure, we have 500 calls. So you would expect making no calls, almost no calls, if at this stage, if we are being more and more conservative, right? But we have 500 calls that we are absolutely sure, no other chance. So this shows that it's not basically a thresholding scheme, but it learns to correct errors. So this, for instance, shows two quality metrics that XHMM uses. Here at this corner, XHMM is also really sure. So if it was a basic thresholding scheme, you would expect all of our corrected calls to be at this corner, meaning that it is not sure, we just filter them out, just maybe like get rid of this corner, right? But as you see, we are also correcting calls that XHMM are really, really sure. So kind of proving our point here. So this is here you see the chromosomes, that human chromosomes, and the length of the reported CNVs in each chromosome. And the greens show the calls that we corrected in this one of the samples. So as you see, we correct calls in all chromosomes and at all varying CNV event lengths. So you might assume that if the length is larger, the method is more confident, but no, actually there are very large calls, relatively larger calls of XHMM that we still correct. And this is the percentage of the calls that we correct. For instance, Y chromosome, which is usually held for the people who analyze this, due to a large number of repeats, we correct 84% of the calls correctly here. So it helps everywhere, but more in certain chromosomes. So, okay, kind of a summary of this. So we were able to consistently improve the precision all around, very fast in seconds in the test time, of course. And, it's independent of the sequencing platform, capture kit, this exome sequencing type, and CNV calling algorithm. So it really works. But shortcomings, we mostly eliminate false positives. So we rarely make new calls. And we cannot make calls if there is no call by the underlying algorithm at this region. So this is a big restriction. And for instance, it didn't provide much help in the cancer dataset. Still low performance on human expert call set. So these were the two limitations, which led us to go to the second stage. And we said, why don't we use the same idea, but have the algorithm of our own? We are basically having an independent copy number variation caller using the same idea and with a different architecture. Yes.
Maybe before going to the second paper,
sure.
I have several questions, but before that, maybe we can ask if there are any questions regarding the first paper.
Yeah.
Are there any? Okay, I have some. Can I ask them?
Yeah, of course.
So I think I understand that regarding the precision, you don't really have a upper or lower bound. You can go up or decrease compared to the baseline method that you're using, for example, XHMM, right?
Mm-hmm.
But regarding the recall, I guess your upper bound is whatever the tool is generating, right? So you cannot really call something that the tool didn't call. Is this correct?
For the recall, our upper bound could be the calls made by CNV or this Chaisen et al. Data, whatever the calls there. So we are supposed to cover them all or not. So yeah, those would be our upper bound, but not the calls made by XHMM or not.
I see. So because what I was thinking is, for example, you're getting some calls from XHMM and then you're correcting them as in, this is not necessarily an CNV, let's say.
That's
right?
true.
But if it is not telling you that there is a CNV at let's say a particular position, you cannot really tell that there should be a CNV.
That's correct. But still you can calculate, consider it within recall, right? Because you know that there has to be a CNV somewhere. And if there's not, it's like a false negative.
Right. So then let's assume that the recall of XHMM is some number, and you cannot probably go above that. But did you check if you're lowering that recall number?
Yes. Okay. Very, very good question. So I didn't report recall at all, and for a very good reason. I mean, for two good reasons. First, it is to the point that, since we are not successful at doing this, so why report recall? So precision is kind of more important, and recall kind of sucks, so we don't even go into that. That's one reason for all methods. The second is, I don't want to go into detail of it, but it is problematic to report recall for this. The reason is, very briefly, the calls that we have on the whole genome sequencing, which is the semi-ground truth, are shorter. And then usually the calls made by exome sequencers are longer because they kind of merge all the calls and then they come up with a longer sequence. So once you have multiple calls made on the whole genome sequencing, which might be contradictory at some points, and then this kind of says there's a duplication in this huge region, and everybody says something else, all the exome sequencers. So it's really how do I calculate a consensus on the whole genome sequencing with these shorter calls? So not really necessary to go into detail, but this discrepancy between the length of the calls and calculating the consensus on these two data types is making it kind of complicated to calculate the recall. And it's hard to also talk about trustworthy and high enough recall with these two. But I'm going to talk in the second one. I'm going to talk about recall because now we have a better way. Yeah.
Are there also other DNM-based CNV callers, or did you have one at least, by the time that you were publishing this paper?
This was the first one that included machine learning this is the first polisher and first method with machine learning, as far as I know. The next one is the first deep learning based method to call CNS, and then there's a new one came from Iman's group. I didn't read it yet, so yeah. So this was the first one.
Maybe two more questions if you have time.
Yeah.
So I think this is a additional step on top of the callers, right? Because there's a calling and then you're sort of filtering, let's say. Did you check what is the overhead of this mechanism in terms of runtime and memory?
It's just seconds.
I see.
Because it takes time to train the model, but after this tool works, XHMM, and then provides us with its predictions and the read depth, it just takes seconds to polish it.
I see. So there is much overhead. Okay. So one more question since I'm curious.
Sure.
Why CNVs? Can't you apply a similar idea on detecting or filtering other?
Like
single methods. Yeah.
Yeah, there are methods doing that, like this deep variant, for instance, deep learning based method. I think, but this wouldn't fit because this works on the read depth signal, and then it has to be a larger region. That's basically the reason. And for translocations and immersions, the read depth wouldn't tell you anything, because once you map it back to the genome, the read depth would still look the same, because the read depth doesn't tell you if the sequence is this way or this way. So yeah, those are the two reasons. But of course, I mean, there are deep learning kind of works for calling smaller variants as well.
Does copy number variation require more coverage to be detected than normal windows?
More coverage required to detect those?
Yes.
So it's better if you have higher coverage, for sure, because the read depth would be less noisy and the method would enjoy having more. Actually, so the second one is under review right now. We are working on revisions, and that's one of the questions: how does the read depth affect the performance of this? And what I see so far is that, as expected, more read depth, better signal, better model.
Also, when you perform in exomes, normally you need to cover more than when you do the whole genome sequence?
More exons than?
In the deep coverage is higher in exomes than the whole genome sequence?
No, no, same. It's usually expected number it would be 30x, like 30 reads per sequence. It goes up to 50x nowadays. So it would be the same. The problem is, because you are restricting yourself to the certain regions of the genome, you are using what's called a capture kit. It is biasing the way you obtain the reads, and the signal is much noisier than whole genome sequencing data. But the depth is similar.
I mean, also when you move to the whole genome to the exome, like the cutting stage where you cut the non-coding part, it happens after or before the sequence machine?
Before. So you tell the sequence machine to kind of to capture only these regions. So you design it before.
Okay.
So that is cheaper and faster.
Okay. So it's not a computational step.
You mean
the cutting?
No. It's not a computation step. So you get the data which is restricted to those regions, and then that's when the computation starts.
Okay. Thanks.
There's a question over here.
So using the associations between whole genome sequencing data and the exome sequencing data, you managed to make some conclusions to enrich this exome sequencing analysis that you do. Could that association that you make or the models that you train also enable lowering the size of data we work with even more than, let's say, whole exome sequencing?
Lowering the size of the data?
Yeah, the amount of data that we work with. So for example, reducing the coverage, or like, I don't know, looking even in fewer regions. Because, so basically my question is like, can those associations that we made with the whole rich data that we had help us kind of make conclusions on even a smaller amount of data?
For this, I think no, because we are not really associating two read depth signals, but we are associating the noisy read depth signal with the call made on that region. So some other algorithm gets to read the nice read depth and then makes a call, and then that information is the only input that goes into our algorithm, our learning method. So
but do you think that would be possible?
Yeah, I think what you, if the question is, can we associate the exome sequencing signal with the genome sequencing signal, which would be something different, but then yes, you can do that. So it could also be possible to predict the whole genome sequencing signal at the same time, but I don't see how it would help us to reduce the data size.
No. So I meant that, let's say that you have this whole exome sequencing, but then it's sequences with some specific coverage from a specific regions, correct? And then that is more limited compared to what we would work with the whole genome
sequencing. Yes.
So I am saying that can be reduce compared to the whole exome sequencing. Can we reduce the regions or like the depth of data we collect from those regions even further, because we have models that train these sparse data to be analyzed more richly based on this. So this, like you said, that there are samples that we have rich data and then like summarized data. So I'm saying that can we even summarize more aggressively?
I think it could be possible. So that, yeah, it is going to be a complicated model probably, but just looking at the exome, maybe you can extrapolate to the nearby regions, and then you don't even sequence those regions.
Right.
Yeah. I think this is doable. I don't know if this would affect the post analysis that people do on whole genome sequencing data, because maybe you are adding some bias or something, but I think this should be quite doable. At least in like germline cases where there is no mutation or affecting something else. I think this is doable. I think it also is a nice idea.
Thanks.
I think this depends on if it makes sense biologically, right? Like if you have a disease that in which mutations are sparse across different types of sequencing, like you find them in exome sequencing, in non-coding regions, or in repetitive regions, then in that sense, you really need to take the whole sequence. But for a disease where you say there's just one rare mutation in a very small repetitive sequencing, then in that case, it makes sense to only consider this small DNA region, right?
Yeah.
So it's very tailored to diseases and
yeah, so that's why I
Okay.
said a germline kind of. If there, I don't know if there's a disease, it might be problematic, but you know, so what we deal with is that these samples do not have any disease. At least that's what that's like considered normal. In this case, you can do that extrapolation, but if there's like a perturbation, yes, then it might be a different story. I agree. Okay. All right. So how much time do I have, John?
15.
15 minutes. Okay, it should be cool. All right. So let's go to the second stage where we said, okay, why don't we, develop our own CNV caller using the same idea and get rid of these limitations that I mentioned. So now what we do is we actually, so this is also on by archive right now, right over here, the QR code. So what we do in this case is that we restrict ourselves to exons right now. So in the, we were considering whatever the call made by this XHMM, and then that was the region we were considering. We say, okay, let's forget about what they do. We'll get the exons, these coding regions, and then make the calls on them one by one. This is also very important because, we also got this in the revision. People are in the clinic interested in sometimes single exon events, just a small exon affected by the copy number variation and what happens. So because it might be like an important exon for certain tissue, which might make all this difference. And then being able to catch those small variations is quite important. So we said, okay, so now we have a nice ground truth. So for every exon, I will have a ground truth. I will get rid of this problem with calculating the recall. We will have the recall, and basically I get the read depth signal over each exon, and I input it into my model. Now we are not using a recurrent neural network, but a transformer model, which I'm sure you have heard. It's been kind of a groundbreaking methods or model, especially in these large language models. So what we do is so we get the read depth signal for each base pair, basically each nucleotide. We map the read depth over that region, which is a single number, into a higher dimensional vector, which is a hyperparameter, 192 in our case. And we do this for every position in the exon, and at most we have 10. So our maximum length is 1,000 base pairs. So from each exon, we calculate a matrix, which is 192 to 1,000, which is the using a fully connected layer. This is like the new kind of an embedding, if you want, like a transformed read depth signal. And we calculate what's called a chromosome-specific position encoding, which is a hard-coded basically procedure. This is not learned. We calculate basically a coordinate for this exon, so that the model is going to know where this exon is within this chromosome. So this is again from the computer vision literature within this transformer architecture. This gives the perspective to the model. So in the recurrent neural network, I knew that the read depth before or after is affecting my decision, my embedding. But here the model doesn't know. The transformer model is not recurrent, so it doesn't know the values of the read depth before or after. It works on self-attention, so it knows which position is important with respect to which other position, and then this helps it, pinpoint the location of this read depth. So we basically, this is learning a fully connected layer. We generate this embedding for this read depth, and using this exome-specific, chromosome-specific encoding, we calculate this and sum them up. Now, one interesting thing here is that I said 192 to 1,000. We add one more vector, which is 192 long, to here, which is called a chromosome-specific classification token. So whereas these are calculated, this is supposed to be learned, and it's specific to each chromosome. So this is like a little token that you attach. The numbers inside is going to be learned by the algorithm as if they are parameters. And this is, as it is going to be chromosome specific, it is going to learn it's going to let us learn the importance of this read depth within this chromosome to be classified as deletion, duplication, or non-com. So we sum these two up, and that's going to be input to our transformer model. So we that's the same thing. So we just sum these two up. We attached our token, and then this 192 to 1001 matrix goes into what's called the transformer architecture. So I'm again not going into much detail. The crucial thing in the transformer model is the multi-head attention module. Basically, it just tries to learn the importance of the read depth on a position with respect to the read depth in the same exon. So it's like an all by all, like a matrix, which learns, this is like exon 1 to exon 1. If I see this read depth over here, what is the importance with respect to each other? So which other read depth values I should look within this exon to focus on, to pay attention? Where should I pay attention? So for each exon, we learn this importance. Where should I look in the read depth? And this chromosome specific token also learns the association or the importance of the this read depth of each exon with respect to each other. So it learns the context. So this is basically like a paragraph, and these are like the sentence, and then the read depth values are the words in each sentence. So I learn the importance of the words with respect to each other within the sentences and in this chromosome, in this paragraph. Yes.
This can be a very naive question, but isn't this like very cell line dependent? Like basically the depth of like each exons, essentially, that you're seeing.
Cell line meaning?
Meaning like different samples, different tissues, let's say. Do we expect to see like different depth of like exons within a single gene?
But we normalize it. And the thing is, within that normalized read depth, if you see some, like a huge peak over here and then something going down here, then it learns maybe that it basically finds out that this sharp decrease in the read depth is important. So yes, it's going to be different for everyone, but the model sees just so many different combinations of read depths for 1,000 patients. And for each exon, think about it, like we have, I think, 800 samples, and then every single exon in those samples is like an input. It's this. Yeah.
And okay, I guess you're not really checking also the RNA-seq
to what?
Yeah, because like my confusion is due to the splicing over there, but probably this is like, you would probably see the similar depth of exons at every cell regardless of the RNA that they are, I guess, generating.
Well,
right?
this is DNA sequencing, so we don't see splicing. We only see if there's some difference. Yeah, that would be, then you do RNA-seq and maybe see it in the tissue.
Okay.
Any other questions? Okay, so we pass this matrix through 3 transformer layers, and it outputs a transformed matrix with the same dimensions. And here's my classification token. Basically, in the transformer architecture, you have an encoder and a decoder. I show you the encoder. So it's like you input an English sentence, and then you are expecting to get the French translation. Then this would have been input to a decoder, which is going to be translated into French with a similar structure. But we are not interested in translating this sentence, if you will, or the paragraph. We are interested in learning something like embedding about this paragraph. And then that embedding is actually stored here in this case. So we basically get this classification token that's specific to this chromosome and use it to classify this this read depth as having a copy number or not. So we take this, it's like a 192 to 1 vector, pass through fully connected layers, softmax, and I output a probability for this to be a duplication, deletion or not. That's going to be my output. Again, the labels come from the same source, whole genome sequencing based semi-ground truth labels. The reason, again, we are using it is quite abundant. I mean, we can do quickly, we have very quickly obtained large number of labels. That's what we need. All right, sure.
Ask it.
I will repeat it.
You need both all the time. So you need to do whole genome and whole exome, to predict for that.
Yeah, so the question is, do we need the both of the data, exome and genome, at the same time? Yes, to train the model, but not when we are predicting at the test time. So, we train this model since we know from this 1,000 genomes consortium, but, say you're a doctor, you do exome sequencing on your patient, you only feed the exome sequencing data to our model, so we predict because we learned. So you don't need the genome sequencing data when you are really using this. What I'm saying is when we are training this.
It's similar. Every copy of structural variation we have among different individuals is very likely to be the same. Otherwise, can't operate in this.
Yeah.
As we go for different populations.
That's very good point. And then I'm going to show you in a second that your point is correct. And yes, it doesn't work if I kind of try it on different set of patients with different set of labels generated by an expert. That's correct.
We also have one question in the chat, but
Okay.
should I read?
All right.
Sure. So Harun is asking this question. He's saying, suppose you were to simulate exome reads from a pangenome and you subjected the ground truth alignments onto each constituent genomes to produce ground truth CNVs. If you were to train using this data instead, how do you think it would compare to training on real data?
So we have simulated over a pangenome. So there are many samples.
Maybe you can also check what's it in the chat.
You cannot do what?
Yeah, I was just going to say maybe.
No, I'm just trying. I see. I'm just trying to understand the question. So we generated reads from pangenome and tried to detect CNVs on it. But why would I want to do that? Since I would like to get predictions for a certain individual to make some conclusions about himself or herself.
Harun, do you want to speak up in case?
Hi, can you hear me?
Yes,
we can.
Yeah, sorry. So the idea is that if you simulate reads from let's say a pangenome, then you already have some let's say structural variation from the different genomes, and then when you surject, it kind of simulates the alignment of reads from a different genome onto another one. Do you kind of see what I mean?
Yeah, but then my point is why, and then the question is, how would it affect the method?
Yeah, as in the idea being that if like, how different do you think the results would be if you were to use simulated data as opposed to real data? As in, I just came up with this as a possible way to simulate this sort of depth or
read that to get more training data.
Exactly.
Okay, I see. I think it might affect depending on how you do the simulation. So if kind of highly resembles the training data we use, it would work. But I'm really I really don't know how the simulation procedure would work, and then how it would change. And then also we don't have a problem of having training data. We don't have a problem of having the training data. We have a problem of having a good set of copy number variation labels, right? But then I also guess we would have the same problem of obtaining good labels on the simulated data. So
yeah, I guess with the simulation, you would know a priori what the exact locations of the reads were. So then you would know like this represents a duplication, or so on. As in, you would have no ambiguity about the labels.
Yeah, I see. Yeah, it would happen. The only thing is that I don't know how the simulation procedure on the pangenome would work. If it is doing a good job of having a similar distribution to what we have, then yes, it might help. But at the same time, it should also be able to nicely simulate the copy number variation read depth. And I'm not, at least I don't know methods that I'm not aware of methods that can do it very well. If there is, then yes, I think it can help.
Okay,
Can help.
thank you.
Thank you. Any other questions? All right. So these are our results. Here I'm showing you the same set of methods that we compared. Many colors, sorry for it, but at the end of the day, this is kind of what matters. And then these are the Polish versions of the same thing. And as you see, the pink one is kind of dominating, other than just the recall on deletion. And then F1 score is basically, now we are talking about recall finally, since we have the ground truth for these exons, and we don't have the problem of merging and then deciding on doing a consensus. As you see, we are kind of the first method to balance the precision and recall to have high performance on both. Actually, we are kind of dominating in all categories. So I call the name of our method is doing a better job than even polishing, because we are now able to make new calls and then they are also precise.
Does it make sense to do a call and decon together?
We thought about this, but I thought not, because if they are working on the same procedure, it might have an effect, but maybe margin. We didn't try that, so we thought it would be. I mean, since the results are good, it's kind of negligible. But yeah, technically we can do the same thing. Maybe we should try it. We thought about it in the beginning and then kind of dropped the idea, but maybe we should again.
Yeah, at least the architectures are different, I guess. At least
that's true. Yeah, that's true. And okay, so this is not important. Again, it works on different platforms. Let's skip this. This is kind of interesting. So with this transformer architecture lets us to visualize the attention weights, meaning, I told you it learns which parts of the read depth signal is important to make a call. So it doesn't basically ignore some parts of the read depth signal. This is not what I need to focus. And yeah, this is what it learns inside, and we can visualize it. So here you see the read depth signal, and then the brighter the color, the more attention it spends. It's like attention is just briefly, when I look at a scene or when I'm talking to you, I'm focusing on your face, and I basically ignore the background. I mean, my attention is on your face, right? So this is where the model looks when making the decision. And as you see in the duplications, for instance, it looks at the parts where the read depth signal is having the lowest value and the highest value. As you see, this is where the focus is going, and same thing for deletion. So it also lets us to visualize and then make sense. This might be a very nice thing to decide on the breakpoints, which is another topic I didn't go into detail. But once you know that there's a copy number variation, since you asked, deciding where it starts and where it ends, it has to be a manual process. And then this is also kind of outputting that, which might be important in a medical setting. All right. So making new calls helped us, and it's nice. Precision recall is all increased. But then again, going to your question, it doesn't work as well when we step out of our training data, which is the 1,000 genomes data set. So this is the cancer data set I just showed you in the beginning. So 16 samples, all validated, and we had a correction. Remember, just one, because XHMM doesn't have a calls anywhere. We had a correction, only one of the samples it had a call. A call doesn't have any calls in any of the samples. So this kind of sucks, because this is a different data set, a different data, cancer data, and yeah, it doesn't work. Similarly, when we calculate the precision, it is around 3% when we do this on the cancer data set, whereas even the count works better, slightly better. So we don't get much improvement as soon as we start to work with labels that are not CMV-nator based. Also, remember we had this 9 samples expert calls, and once we test the model on these calls that the human expert did, our F1 improvement is just very slight compared to others. So it really doesn't provide much improvement. And here, for instance, the precision is 1, but they don't have much recall here, as you see. So we are basically second, third, marginal improvement in both, so it doesn't work well. So can we train an recall model using these data sets? One has 16 samples, one has 9 samples. The answer is no. because it's so small to train such a large complex model. But we can fine-tune the model. So fine-tuning is basically you train your model on a large-scale data with this semi-ground label, so it learns something. Maybe not the best thing, but it's like a general-purpose training, if you will. Now, if you want to specialize on a certain data set, but you don't have much labels, small data set, then you can use the trained model, transfer it, basically get the weights that the learned model, change it with these samples, but just a little bit, called fine-tuning. So we can fine-tune the model using these samples. So what we do is we have a new model called call fine-tuned expert. So we had 9 samples from the human expert. Just fine-tune it with 4 samples and test on the remaining 4 or 5. And similarly, train with 3 samples of the cancer data set, have this somatic version, and test with the rest of them. And let's see how the results change. So on the cancer data set, now, so we trained with these not available ones, not applicable ones, and in all of the rest, with the somatic fine-tuned model, we are able to detect these copy numbers. And now our precision over our F1 was 3%. Now with the fine-tuned model, it's around 30%. Still not the best, but much better than 3%. Than that kind of. You get the point. And then these are the results of the FT expert model on these, human expert labels. Once we fine-tune with some of the samples that they provide, it's the first time, I guess, we are able to reach like 60% F1 score. And again, the precision and recall is high at the same time, which is also very desirable. All right, so copy number variation is challenging on whole exome sequencing data, and we propose 2 solutions. And with a call model, we have the best precision and recall both high at the same time for the first time. So this is a promising direction to use exome sequencing data in clinic, but yeah, there's still some room for improvement. So one other advantage is a germline, meaning normal tissue, and somatic, meaning tumor tissue caller. Usually, people have different versions of these callers to compare and contrast. And you need both, for instance, for a cancer-based somatic caller, you need both the normal tissue and the tumor tissue. So you have to sequence both, double the cost and stuff. So we don't basically need that, as we had shown. You're only using the cancer data, you are able to fine-tune the model and come up with a bladder cancer model to make calls. So that's all. Here are my QR codes if you are interested. And thanks to our funding agencies, you can reach me here, and I'm happy to take more questions if you have. Thank you.
Thanks. So since there's a delay on YouTube, let's at least wait at least 10 or 15 seconds to see if there are any questions. In the meantime, yeah. If there are questions.
Should I take that?
Yeah.
Yes, my question would be like, what is the other benefits on doing exome rather than whole genome sequencing?
Exome sequencing?
The benefit of doing exome rather than genome sequencing, because I think based on the method that you just explained, you still need to go back to you need to have both. And also to determine the sequence of what's really going there, you need to go back to the genome sequences. Seems like if you just do whole genome sequence, I think maybe just the precision of determining the variation that are there is the only benefit.
No, but you don't need to you don't need both. You only need exomes to do exome sequencing, because luckily these consortiums have done both exome and genome for thousands of people. So we are able to use that data to train the model. But once the model is trained, it would only accept exome sequencing data as input. So if you're a doctor, you can only do exome sequencing, input it into our model, and obtain the performance you would obtain using the genome sequencing data, and maybe even more. So that's the point. So genome sequencing is costly, takes more time, takes more space. Exome is widely used because it's the reverse of these. So we are kind of taking the burden out of doing whole genome sequencing.
Okay.
maybe two more.
Sure.
I feel like these are already sort of mentioned, but just to clarify. So when we have, let's say, another type of larger data sets like 1,000 genome, right? Because like the claims are currently, we don't have like other comprehensive data sets, the JSON data set, I guess, right? The sample number is pretty low. So do you think, again, like we would see same accuracy numbers, like as you shown with the JSON data trained on the 1,000 genome? If that's the case, do you think the solution is always fine-tuning to a particular data set? And maybe related to it, what is your validation method? Is this cross-validation, let's say you train on one set of data and then infer now really unseen data, again, using a 1,000 genome?
Yeah, so you say if you have larger data set than 1,000 genomes?
Exactly.
Yeah, it would be the same. The problem is obtaining the calls. So if we have like a army of people, have validated calls on these 1,000 samples that we use from 1,000 genomes data set, we would have better results. Unless we have a way of labeling these, by humans, by clinical experts in a faster and more accurate way, the larger data set is not going to have. We are not even using the full 1,000 genomes data set, like maybe half of it at most. So it's not going to help. The problem is having the labels. And for the second question, yes, what we do is we have 9 samples. We take 4 of them to fine-tune the model. We have 4 unrelated samples. We test on those. And same for the cancer one. But then the model learns the decision-making process of the human expert on those 4 samples and kind of directs it towards the other ones. But first you need to train, like have a pre-trained model, which had already learned about copy number variation using a large number of samples, like we use 800, I guess. And that's only possible with semi-automated calling. For this, we use CNVs.
Makes sense. So one last question, maybe very high level. Then I'll give to you, Nico. So what do you think the future of whole exome sequencing is? If you assume that the whole genome sequencing is getting a lot cheaper and the storage costs are either reduced or they are not as important as—
You mean genome or exome?
If the cost of whole genome sequencing is being reduced.
You said exome.
Yeah.
Maybe I got it. Okay. Yeah. So this is a good question. So what happens if exome sequencing is used at some point, and then we use genome sequencing, and then as genome sequencing costs are getting lower, then it's a feasible thing. And still, we are going to need this. So we are talking about this with a group in San Francisco, and then they are doing, they kind of switched to using whole genome sequencing in their lab, but still they are seeking solutions for higher sensitivity in certain regions, because even using whole genome sequencing, because of the repeats in the genome, just you don't have good performance on certain regions. And if it is the gene of your interest, then you still need some method which is going to fine-tune for that. So we are actually thinking about having this idea working on whole genome to whole genome kind of version. Yeah. So I think that if right now it's still whole exome sequencing is an important resource. If it comes to that, then still I think at whole genome sequencing we are going to need such methods.
Even with very long and very—
depends on the read depth with respect to the long reads. There are methods I see now that kind of combines them, and then using long reads. Long reads can help actually, but then I think we still need to read depth as well.
One last question.
So you mentioned about providing context in the sense of chromosomes so that you can enrich the predictions. So I'm wondering if it is possible to give even broader context or more fine-grained context. So broader, for example, across chromosome or even across population at a specific region, or like people with the specific features. Would providing that broader context help? Let's say that we want to analyze some issues like a neurological issue, and then we realized that there was some people who have this age, or not age, sorry, but like from this part of the world, or like have the other type of, let's say, condition that will provide a context to enrich the analysis of, let's say, data in this specific case.
Good question. So to answer your first question, we tried. So we tried this without this chromosome-specific token, meaning we had the token, but it is kind of shared across all chromosomes. We had worse results compared to this, because if you think about it, if you have a copy number variation, and then if you see a read depth signal change in some certain region, I would expect it to be more related to the read depth change around that region, and next, maybe like a larger neighborhood, but in that chromosome, not in other chromosome. That's what I would expect. So if you have the duplication or a deletion in a certain region of the chromosome, I think it should be localized. And then we had better results by, kind of having this token specific to chromosome. So that's why we went for it. So yeah, you have a follow-up?
Yes. So that makes sense. Thanks. But then the question here would be that, let's say we want to localize it across a specific chromosome, but for, let's say, people that have liver cancer. So imagine that we have trained a model of this specific chromosome for people with this condition, and then we want to analyze another condition. So let's say whoever has liver cancer might also have some other condition. So I'm just providing a non-informed example, but
I get your question.
Yeah.
So one other thing is, even though we have this specialization, it doesn't mean the model is learning it doesn't share anything across chromosomes. So for instance, so remember we had earlier fully connected pairs, even though this classification token is chromosome specific, the loss that we calculate over the neural network will affect those layers. So, and then it's shared across chromosomes. So it's not really totally independent. For instance, we tried models where we only train it on for a certain chromosome. And there are certain chromosomes which, really our model do not perform well because of the problems probably that on that chromosome. But since we are doing it, when we do it for all chromosomes, then for that chromosome, we have better results. So this is showing they are not totally isolated. So we are learning, even though we have this specialization, which improves our results, they still share information, which is necessary for broader results. As for, so it kind of answers your question about having this specific condition. I think there what really matters is showing the model with samples with that condition.
Yes.
Yeah. So that's usually the like our bladder cancer case. We have only 16 samples. Not much, so you cannot train a model from scratch.
Okay.
But you train this model, and then maybe with a few samples, you kind of change the weights a little bit, fine tune it. Then it learns to kind of focus more on this special case.
Okay, I guess if there are any other questions, we'll have to take it offline. So let's thank our speaker again. Thanks a lot. Unless there are any other comments or anything from the Zoom audience, we'll close the session. Okay, probably there aren't.